Welcome back. In the first lecture, I gave you a sense of what neuroscience is, the discipline of neuroscience, and how neuroscience touches our everyday lives. And now that I've given you a feeling for the discipline, what I'd like to do is start to give you a feeling for how a neuroscientist studies the brain, exactly what it is that we do, and what kinds of explanations we're looking for. So what I'd like to do is to give you that feeling by walking you through some ways in which we neuroscientists look at the brain. And in particular, I want to show you how a neuroscientist tests an idea about how the brain works, and also at what level we explain a phenomenon. Now, let's look at these levels of structure. Neuroscientists are interested in the function of the nervous system at different levels of structure. We are interested at levels ranging all the way from molecules all the way up to the whole brain, the whole central nervous system. And what I want you to think about is the fact that there are different levels of structure, all of which are of interest to neuroscientists. And if you think about at the lowest level, individual molecules, so we're talking about neurotransmitters, we're also talking about ion channels, things that we'll be hearing about as the molecules that constitute neurons and those molecules that work together to make how a single neuron works. And if we go up from those molecules, then we start getting to structures such as synapses, which are the connections between neurons. And those synapses are the connections that neurons use to talk to one another. And then going up from synapses, then we get to neurons. Looking at neurons, then these are the cells of the brain that are responsible for sending electrical signals over long distances. And then building up from neurons, then we can build up from neurons up to networks. Networks are groups of neurons talking to one another. Maps, the brain is filled with maps, and so in fact a central principle of brain organization is the mapping of information from the world into the brain and also through from one brain region to another. And then going from the map level, then going up to systems, so for instance an entire network of brain regions that talk to one another to generate a behavior, and as I said, all the way to the highest level, the central nervous system, the entire brain. These are all different levels of structure that we can investigate experimentally in animals and sometimes even in humans. Now, another way to look at brain function is in terms of the mechanisms that generate brains, that generate changes in the brain throughout life. And these mechanisms begin with genetics, so the endowment that we receive from our mother and our father when we're born, and even before we're born as we develop, the developmental steps that lead to the formation of a brain, and those steps that lead our brains to develop, to grow, and to become mature brains. And that's another type of process that neuroscientists are very much engaged in studying. Of course, the environment has a role in shaping who we are, and the environment, by that I mean plasticity, experience, things that happen to our brains as we develop, and even in adult life, things, events that shape our brains and make us who we are. And finally, neuroscientists are of course interested in behavior, and now this touches on what I mentioned in the previous lecture, phenomena that are the province of psychology, of psychiatry, sociology, and even economics. And so these are all levels of explanation that are of interest to a neuroscientist. Now, these approaches aren't mutually exclusive. Often a scientist will be interested in investigating, say, genetics and maps, or perhaps behavior and development. So when I present you this list of approaches, it's not that they're mutually exclusive. They're just different ways of outlining, in broad strokes, the kinds of explanations that neuroscientists look for, the kinds of things that we want in understanding how the brain works a little bit better. And in all cases, the key is to have some possible explanation in mind. There's some model for how the brain works. And what we have in mind is some model that we'd like to test with an experiment. And the idea is that we have this model, this general conceptual framework, and we design an experiment, we do the experiment, and if the experiment gives a result that matches our prior views, then the idea that we have gets to stick around for a while. If that's not the case, then it's time to find another explanation. And so constantly science is composed of these steps of forming a hypothesis, forming a worldview, and then going and making a measurement, and then seeing how the measurement turns out, seeing how the experiment turns out. So that's the process of scientific discovery, and that's the process by which thousands of neuroscientists have discovered the kinds of things that we'll be talking about. Now, let me give you some examples of experiments that neuroscientists have done, examples of evidence that we have for how some particular part of the brain may work. So here are a few examples. First example, we could examine a patient who has had a stroke in some part of the brain, and we could see whether he or she had some deficit in function, for instance, a deficit in speech, or a deficit in the ability to, say, see a moving object or to see color. These are examples of deficits that can arise from strokes, And they tell us whether some particular part of the brain is important for some function, whether it be speech or seeing color or even higher reasoning or exercising self-control. Those are all examples of what neuroscientists call lesion experiments, in which you can look and see either in a naturally occurring case or in an experimentally induced case, in the case of an animal, what goes wrong when some part of the brain is missing. So that's a whole category of experiment that people do. Here's another example. This example involves measuring activity in part of an animal's brain. And so in this example, what we would do is we would measure activity in some part of an animal's brain, or perhaps we'd measure the level of some chemical and see whether that chemical or electrical activity is there when the animal is doing something. So for instance, if an animal hears a sound or is running in a maze. And the idea here is to look for things that are neural correlates or some kind of correlate of a behavior. And the idea would be that a neural correlate is something that's a candidate for something that might generate that behavior, something that might be critical for that behavior. And then if we wanted to, we would then find a way to remove that activity or chemical and see if that event, that electrical event or that chemical signal, were necessary for generating the behavior. So that's another kind of experiment that one might do. Finally, third example, we could look for a genetic difference. So for instance, if we were interested in finding out whether some behavioral traits such as autism, which is, as I mentioned before, of developmental origins, we might want to look for a gene that underlies autism. We might want to look for some gene that is different in some group of people, such as autistic people, versus people who are not autistic, typicals. And so these are all examples of things that we might do, experimental approaches we might take for understanding brain function. Now, as I've given you these three examples, let's think about this a little bit. Each one of these approaches is at a level that tells us something a little bit different. So for instance, in one example, I said maybe we want a molecule or a gene, and then we understand that that gene is somehow involved. Other times I mentioned that we would want to record from neurons, or perhaps we'd even want to image a whole brain. And what's important to remember is that these different levels of explanation give us something important, tell us something about brain function, but they're not necessarily going to tell us something about another level. So ultimately, what would be nice would be to come up with levels of explanation that join these different levels of analysis. For instance, joining molecules to synapses, to neurons, to circuits. And so one general goal in constructing these scientific explanations is to come up with some general framework in which these different levels of explanation can start to connect to one another. And I haven't even mentioned other ways of doing experiments or explaining data. For instance, we might look for an explanatory framework in terms of evolution. Natural selection is the process that's generated all life, including the evolution of the brain. And so we might want to look for some evolutionary perspective for how it is that some behavior arose. Another source of great insight would be psychology. A great stream of research that's given rise to modern neuroscience is psychology, in which people studied behavior and mental function without invading the brain, just by simply observing people behaving. And that whole discipline of psychology has been addressing a lot of the questions that we're going to be talking about in this course for over a century. And so this is a rich source of insights and perspectives for understanding neuroscientific explanations. And taken together, all these different kinds of explanations can lead to deeper insights into behavior. So, now, all these different approaches, all of these different levels of analysis call for different kinds of experiments. And what experiment we do depends on exactly what it is we're trying to find out. And so designing an experiment can roughly be thought of as being a three-component process that leads to a particular experiment being designed or done. So, the first step would be to think about what brain region and even what animal in which to test the idea. Now, when I say what animal, this is what I mean. A central discovery over the last 100 years of neuroscience is that the vast majority that our brains do are found in other animals. For instance, sometimes in monkeys, rodents, and birds. And so I'll give you examples, for instance, basic sensory transduction, how a touch gets translated into an electrical signal, or perhaps how we perceive color, how we perceive motion, even how we estimate the number of objects in the field of view. All of these things are things that animals can do. And so it's possible to find an animal model for doing these things. And that's very important for neuroscientific explanation. And this extends even to very low levels, because it's not just monkeys, rodents, and birds. We can often find animals that have the things that we'd like to study in worms, flies, and even in squid. So for example, the fundamental basis of the action potential, the fundamental unit of electrical signaling in the brain, is something that's shared between us and squid. And these deep principles are shared among a wide range of animals, all the way from squid up to us. The universality of these deep principles of brain function allow us to discover principles of brain function that are shared across a wide range of organisms. And what that means is that there's a hope of studying animals in order to understand human behavior and sometimes human disease. And so the first step is then looking for an animal and looking for a particular brain region or a phenomenon in an animal to study. Now, another step is to think about what hypothesis is suggested by the existing evidence. So, for example, we might look in the literature, we might look at existing experimental evidence to see what exactly it is that we're trying to study, see what's already known, and what's known might suggest that a particular experiment is worth doing. So, for instance, knowing that there's a gene that's involved in early development, such as in autism, might suggest that we should start looking in early stages of development and look at developing brains, and that would be the next step. So an existing hypothesis then would suggest that a particular experiment is worth doing. And then finally, there's a simple technological issue, and this technological issue is at the forefront of neuroscience. Is it technically possible to ask a question at the level you'd like? And this last step is a very important one because it's an exciting part of neuroscience today that technologies are being developed to help us understand, say, for instance, the molecular biology of the brain a little bit better, or the development, or the signals that happen in neural circuits as an animal or a human being is thinking about something or is processing information. And so this technological issue is very important because the technology limits what it is that we're able to do, in terms of an experiment. Now, what I'd like to do is give you a few examples today, 2 examples to illustrate these principles and to show you exactly how these principles come into play in these simple examples of beliefs that you may, in fact, already know about. And the examples I want to give you are 2 ideas that are commonly held beliefs. The first one is the idea that alcohol kills neurons. Okay, that's a common belief. And the second one is the idea that classical music somehow makes babies smarter. Now, in each case, there's something interesting and relevant to our lives, so it's something that I hope I can convince you is relevant to your everyday life. But also, I want to show you what these questions, again, look like from a neuroscientific perspective and at different levels of explanation from a neuroscientist's point of view. So, the first example is alcohol and alcohol killing brain cells. It's commonly the case that at parties we make jokes like, ho ho, bottoms up, we're killing brain cells here. And that's the kind of thing that's very embedded in the popular culture. People think that they are killing brain cells when they take a drink. Now, where did this belief come from? Well, the original finding that led to this belief is that when heavy drinkers were examined post-mortem, it was found that heavy drinkers have smaller brains. Their brains were smaller. And so, well, if their brains are smaller, well, brains are made of cells, and those cells are neurons. So therefore, it must be the case that drinking kills neurons, right? After all, most of the brain space is filled with neurons, and so if it shrinks, there must be fewer neurons, right? And so that's how the belief was born. However, there is a loophole. And let me just show you what neurons look like. So this is a neuron. Now, the brain is made of neurons, as I said. In particular, this is a neuron that I filled with fluorescent dyes so that I could see it a little bit better. And this is one that I took in the laboratory and made this image that we're looking at here. Brains are made of neurons. And brains are closely packed structures in which there are many neurons packed cheek by jowl. And the reason that we can see this neuron is that I filled it with fluorescent dye. And therefore, we can see this neuron, but not the other neurons that are next to it. So brain tissue is made of neurons that are closely packed next to one another and filling space. Now, the loophole here in this idea that somehow drinking kills nerve cells, kills neurons, is that neurons don't have to stay the same size. If we look at this neuron, it's quite apparent that this could change in size. What we're looking at in this nerve cell is a cell body. That's the thing that the pipette is sticking onto. So I filled it at the cell body. Now, if you go up from the cell body, there's this large, complex tree, this almost rococo structure that's basically filling much more volume than the cell body itself. And that's called the dendrite. That's the input of the neuron, and that's where inputs come into the nerve cell, and then the nerve cell integrates the input. Okay? And then there's the axon. That's the structure that comes out of the bottom. That structure that comes out of the bottom is the output of the neuron. Now, the point here is that this neuron is composed of many components. And because of that, this neuron can change in volume. If you think about that, that means that, well, maybe there isn't a loss of nerve cells. Maybe, in fact, the cells are just getting smaller. So there's another bit of evidence that's been gathered. And the bit of evidence is that anatomists have counted the number of cells in brains of heavy drinkers. So instead of measuring mass, what they did is they used a microscope to see neurons directly. And what they did is they took a piece of tissue and they figured out how many nerve cells there were in the tissue, and they used that to then estimate how many neurons there were present in total in the entire brain. And what they found was that the total number of neurons in the brain of heavy drinkers is not reduced, okay? And so what that shows is that heavy drinking apparently, at least according to these measurements that were made, does not have an effect on the number of neurons. And what that suggests is the possibility that individual neurons are shrinking. Perhaps the dendritic arbor, this large structure that I just have been showing you, perhaps that dendritic tree gets a little bit smaller. Now, that's a speculation. That particular speculation that I just made has not been tested, to my knowledge, but it could be tested by future experiments. And I think you can see how it would be tested. It would be tested by examining these neuronal arbors. Now, before you get your hopes up about drinking and neurons, it is not necessarily the case that drinking is good for the brain. Now, let's get into this a little bit more. Now, first off, can neurons ever be killed by heavy drinking? Well, as it turns out, yes. It turns out after decades of drinking, there is a syndrome called Wernicke-Korsakoff syndrome. And this is a syndrome in which drinking leads to actual loss of brain cells. It turns out that the way this works is that people drink so much that they get all their calories from alcohol and develop a thiamine deficiency. And that thiamine deficiency then causes neurons to die. So that would be one way in which neurons can be lost. Here's another way that neurons can be lost. In the developing brain. If you look in the developing brains of fetuses, new neurons are being born all the time. Okay, this is part of brain development. And in a pregnant rat, even the equivalent of one drink can lead to measurable slowing in the birth of neurons. So during pregnancy, drinking can most certainly lead to reduction in neuron number. Now, let's think about this a little bit. What consequences are there for everyday life? So one consequence is that if neurons can shrink, well, they might be able to grow again. And in fact, it's been shown that giving up drinking is associated with recovery of brain mass. So one possibility is that even if you're a heavy drinker and have experienced loss of cognitive function, it might be possible that you could, in fact, recover some brain mass if you were to stop drinking. Now, another consequence, based on literature evidence, is that, in fact, small amounts of alcohol may be safe. Moderate amounts of drinking are not shown to cause any measurable amount of tissue loss or of loss of function. And in fact, there is an amount of drinking that's been demonstrated to be safe, and it's generally regarded as being safe to have about 2 drinks per day for men and about 1 drink per day for women. So that's a generally regarded safe dose of alcohol in the diet. And finally, there's a very specific exception to this. Not only is alcohol innocuous in moderate doses, in fact, some alcoholic beverages can, in fact, be somewhat beneficial, somewhat protective of brain function. And in particular, red wine is not neutral, but slightly beneficial. There's been a demonstration that drinking red wine seems to be protective. It seems to preserve cognitive function, especially in older persons. So some moderate amount of red wine may, in fact, preserve cognitive function. Now, I should say that one of the studies that demonstrated this was done at the University of Bordeaux. Now, that doesn't rule out it being true. There are studies that are also done in non-wine producing regions that, in fact, also show the same thing. Now, second example, and I'll call this second example the Mozart myth. The example goes like this. Playing classical music to babies or even to fetuses, say when they're in the mother's womb, can lead to later increases in IQ. That's the belief. And this is a commonplace belief. It is reflected in products that you can see on late night television. For instance, a product that's called Your Baby Can Read. And so the product goes like this. If you give really early enrichment, you can show babies really complicated things and they will learn them. Another example is so-called baby DVDs, in which you can show cartoons of geometrical objects and classical music and play these to infants, and somehow this is going to make them smarter. Now, where did this come from? This belief originated in a technical report, and it originated in a technical report in a leading scientific journal, specifically Nature, which is a leading international journal of science. And the original finding was that playing classical music to a group made them somewhat better in one of a group of intelligence tests. Now, the group of people was not babies, as it turns out. The group of people was college students. And so the first difference between the belief and the result is that the original result was obtained using college students. The second difference between the popular belief and the original observation was that the original observation was a transient effect lasting about 20 minutes. So in half an hour, the effect was gone. So these college students did better on one test, and then after about half an hour, then their performance was back to what it was before they were listening to Mozart or Bach or what have you. Now, that was the original finding. Now, it turns out that there was a transition that was made. Originally, when this report came out, it came out in the popular press. It was the subject of news reports saying that college students could do better when listening to classical music. But gradually, those reports from college students were replaced by other reports saying that it was babies who benefited. Now, what happened in between? It turns out that what happened in between was the emergence of a popular book called The Mozart Effect. And this popular book had no new evidence, but it claimed that classical music could do all kinds of things. Now, it's ironic that that led to a transition of the Mozart effect applying to first adults and then to babies, because in fact, the original result has subsequently been refuted. There have been more studies done, and it turns out the original result was not reproducible. More studies have been done, and as is sometimes the case in scientific literature, an original finding doesn't hold up, and it's no longer possible to observe the effect in the laboratory. So it turns out that there is in fact no evidence for this so-called Mozart effect. And so if we think about products such as Your Baby Can Read, this idea that you can teach an infant to read, or perhaps the idea that playing videos of geometrical shapes with classical music to infants can make them smarter, these ideas are not true. And in fact, even one of these products, it turns out, was advertised under those circumstances, and now you can get your money back after a lawsuit. So it is possible to get your money back from some of these products that have claimed this quality of classical music being played to babies. Now, I should say that this could have been a neuroscience question. This could have been the subject of further investigation. It could have been the case that that playing classical music would be something that you might want to do to, I don't know, a dish of neurons. But one thing that was missing here was a coherent explanation for how it is that passive listening to music could somehow have some kind of effect so early in life. Because as far as we can tell, babies don't really have the neural structures to really perceive these complex structures in music. And what's known about music perception is that it's something that's acquired later on. Now it turns out that it's not all bad news. It turns out that despite the fact that there is no known benefit of playing classical music to infants, there is something about music that does seem to improve performance. And what is known is that active production of music seems to be correlated with better performance at spatial reasoning tasks. And in particular, when children learn to play a musical instrument, that seems to be correlated with better performance at spatial reasoning tasks. And that's something that demonstrates the principle that the active engagement of the brain, the active learning and practice of skills, does seem to have some interaction with the acquisition of spatial reasoning abilities. And so there is some kernel there that perhaps can be applied to everyday life, but it's not the idea that's in the culture of playing Mozart to babies. Now, I've given you these two examples, one of which is the baby Einstein effect and the other, the Mozart effect, and the other is alcohol in nerve cells. And what I'm trying to give you here is a sense of how scientists apply their desire for explanations at different levels. And let me summarize what we've learned here. What we've learned is how a neuroscientist looks for explanations. We've learned that there's a scientific method for investigating questions. Ideas have to be tested to be considered scientific. There has to be a hypothesis. The hypothesis has to be formed. An experiment has to be designed. And then the experiment tells you whether the hypothesis can stick around a while longer or whether the hypothesis is in fact apparently wrong and needs to be dropped. And so a central part of the scientific method is that any idea is provisional. The idea is there. It can survive for a while. But if a piece of evidence comes in that contradicts the idea, then the idea has to be tossed, and we have to go back to the drawing board and think about what other idea that we might entertain. Another component of scientific explanation is what I mentioned at the beginning, which is that levels of explanation have to connect with one another. So I mentioned that one might think about synaptic function in terms of molecules or cellular function in terms of synapses. These different levels of explanation have to connect with one another. And eventually what neuroscientists are looking for is some large framework of explanation. These large frameworks need to have ideas at different levels that connect to one another. So one might eventually want to come up with an explanation, for instance, for Alzheimer's disease that is rooted in the genetics of Alzheimer's disease. And these large frameworks are a long-term goal of neuroscientists. And this long-term goal is to come up with these frameworks. And once these frameworks have stuck around for long enough, once they've started maturing at different levels, we start calling these frameworks theories. And that's a long-term goal in neuroscience.